to introduce Dr. Marek Kubicki as our speaker. So Dr. Marek Kubicki is a professor of psychiatry at the Brigham and Women's Hospital, Harvard Medical School, and a trained radiologist holding an MD and a PhD degree from the Medical University of Łódź, Poland. He holds multiple appointments. So he has his primary appointment with psychiatry at Brigham and Women's, his secondary appointment with the Department of Radiology, both at Brigham and Women's, and the Department of Psychiatry at Massachusetts General Hospital, Mass Gen. So he's a co-director of the Center for Morphometric Analysis, MGH, and the associate director for the Psychiatry Neuroimaging Laboratory, Brigham and Women's, which he also helped co-found. So PNL at Harvard is a very famous lab, everyone knows it. But I know of Marek's work, and this is actually the first time I'm meeting him. So I know of Marek's work from his association with the Laboratory of Mathematical Imaging, LMI, another famous lab at Harvard. So this lab is one of the very few places in the world I know, which is a very nice concentration of applied mathematicians, statisticians, computer scientists, engineers, clinicians, and neuroscientists all working together. And that's how I know of Marek, not from his work in psychosis and psychiatry, which of course he's very famous for, but I know his work from methods. So throughout his career, he's been awarded many prestigious awards, including the NIH Young Investigator Award, 2 consecutive NARSAD Young Investigator Awards, ACNP Young Investigator Award, and the Harvard Milton Award for consecutive years. So the Harvard Milton Award, it's from a Milton Fund, which is one of the oldest bequeathed funds at Harvard. So he's very heavily involved in teaching and mentoring, and so he's graduated lots of postdoctoral fellows, trainees, and other fellows, research trainees. So he runs, he co-directs the T32 training program at Harvard, the Stuart Hauser Clinical Research Training Program in Biological and Social Psychiatry, and of course is himself funded from a K24 mentoring NIMH award, rightly so. So he's on the editorial boards of various journals, Schizophrenia Research, Psychiatry Research, Neuroimaging, BNC Psychiatry, Brain Imaging Behavior, and is also the founder, editorial board member of Journal of Neuroimaging, Psychiatry, and Neurology. So all of these journals we have also published papers in, although now is not the time to confront him about our reviews. So Marek is at the forefront of applying and developing cutting-edge diffusion imaging methods to schizophrenia, and these are now being widely used to understand human brain health and disease. So his talk today is going to be on, so he changed the title, I see, so it's going to be about imaging in neuropsychiatry from big data to biomarker development. So since I know Marek's work from methods and morphometry, and he's been on papers with several of mathematicians whom I respect and I look up to, he could actually give 3 or 4 separate talks, both on image analysis, data harmonization, application of these diffusion imaging methods to psychosis, all 3 separate talks. You left out acquisition, which is also 4th talk, all of which I would gladly sit through and listen to. But today he's going to summarize all these things and give us a concise finding of his recent research. So please welcome Marek.
Oh, thank you so much for this introduction. I didn't know that much about me. All right, so thank you for inviting me, Shantanu and Katrin. I'm glad to be here. As Shantanu said, I have been for the last 20 years involved in this research where we're trying to understand we're working on developing methods, tools, measures, and data analytic pipelines to different neuropsychiatric disorders. Majority of our work is in diffusion imaging, diffusion tensor imaging, and we are applying it to Mostly schizophrenia, but also other diseases. We've been working with traumatic brain injury data, with depression data, with autism, with all sorts of other disorders. Today, I will be presenting two separate methods or tools or approaches to analyzing the data. In the first part, I will focus on what can be done with large data set or set of different data sets acquired in different magnets, different scanners, different places, and how to go about combining them and analyzing them through increasing the sensitivity and power of the analysis. Then the second part of my talk is going to be about how to use diffusion imaging to increase specificity or to understand better underlying biology behind disease like schizophrenia. For those that are not familiar with schizophrenia, since majority of the talk is going to be about this disorder, there's a small sort of summary of what is and what we know about it. It's a devastating disorder that affects just under 1% of population. The onset of symptoms are in late adolescence or early adulthood. It has a clear genetic risk. It is considered a developmental disorder, but shows progression, and we're still not sure what actually causes the disease and what happens during and along the time course of schizophrenia. The reason why we are focusing on white matter is at least threefold. For one, schizophrenia has always been believed a disconnection syndrome. Majority of the clinical symptoms are arising from disconnectivity or fault connectivity between frontal and temporal lobes. It's been proposed already in 1906 by Wernicke. It has been reinforced by studies by Friston, Weinberger. They are mostly functional studies. However, there's also a structural, microstructural evidence for myelin abnormalities. Myelin surrounds the axons and provides communication between distant brain regions. There's also postmortem and DNA evidence for myelin abnormalities. And since 1994-95, when diffusion tensor imaging came to life and to clinical applications, many people has been using it to test this connectivity, disconnectivity, myelin hypothesis in schizophrenia. Diffusion tensor imaging, a lot of you are using this technique. Here, it is a very powerful technique. It can visualize or can help visualizing white matter connections. One can not only detect and characterize water diffusion at the voxel level, but also connect those voxels that seem to be visualizing white matter myelinated axons, and if you connect them, then you form this line that is called tractograph or tract that follows the axons from one gray matter regions all the way to the other gray matter regions. At each location, diffusion behavior of water is modeled as ellipsoid, and we called it a tensor. So all those ellipsoids are called tensors. You can see that they have different shapes. That is simply because the water diffusion behavior is different, and the voxel here, where water cannot move perpendicular to the axons in white matter. This is corpus callosum, versus regions like here, like a CSF, where water can move in all directions, and this diffusion tensor is very spherical. It's even better described here. We are talking about this diffusion anisotropy, so whatever water can do in this one voxel, how it can move, can be visualized and then quantified by diffusion tensor. And this tensor has 3 perpendicular directions, eigenvectors, and these eigenvectors have eigenvalues. This is a little bit mathematical, but what it means is that if in the area where all the axons are organized like this and all the fibers are parallel to each other, there's very little space for water to move perpendicular to the axons. It can move only along the axons. Right, so this anisotropy is going to be this tensor is going to be very flat and very long. We measure it through a measure called fractional anisotropy. This is one of the measures, the most popular, and this, the longer this ellipsoid, the higher fractional anisotropy. Unfortunately for fractional anisotropy and for our research, it is very non-specific. It's very sensitive. It's sensitive to almost everything that happens in white matter, but it's very non-specific. Axonal density can change anisotropy. Myelination can increase anisotropy. Demyelination can decrease anisotropy. Whenever there are fiber crossings, the anisotropy goes down. The expansion of extracellular space through the edema and inflammation expands the extracellular space, decreases anisotropy, and glial scars also change anisotropy and change them differently in gray matter and white matter. So all these problems, fractional. So we have the method that can visualize and quantify white matter in schizophrenia. We can now apply it to test this myelin hypothesis. Unfortunately, because of all these problems of non-specificity, we have non-aligning findings. The literature is producing a lot of papers. Those papers are not really showing the same thing. They show abnormalities, but they show abnormalities all over the place. This was one of the little exercises that one of our summer students did in 2010 and published in 2011, where we asked him to find all the papers that were using voxel-based whole brain analysis and plot them in a common space, in MNI space. We're hoping to find some kind of hotspots where schizophrenia would be, affected in white matter. The candidate regions were obviously frontal and temporal lobes on the left side and on the right side. But it doesn't seem like this is the case. It seems like both first episode and chronic schizophrenia, like red is chronic, yellow is first episode. They show abnormalities all over the place. Each paper reports something different, and they're not overlapping with each other. They're not replicating each other. One of the principal reasons for this is that neuroimaging studies are very expensive, and doing these studies requires a lot of money, a lot of funding, and the studies are usually based on very small sample sizes. So they are based on 30, 40 patients, a majority of the published studies, and they might be underpowered. The diffusion data is known to be noisy, so the power of this analysis might not be enough to consistently find this pathology. So obviously things like looking at trajectories of white matter with age and with disease progression are also impossible to do with this limited cross-sectional small samples. Around the time where we published that study, a few years later, Peter Kochanov published this very impressive study where they were sort of combining together all the hypotheses that would be nice to test with diffusion imaging. The white matter obviously seems to be affected in schizophrenia. It seems to be myelination, but how it interacts with maturation and how it interacts with the onset of psychosis is of great interest. And another reason for looking at white matter, at least based on this paper, is that the white matter maturation peaks around the psychosis onset. So not the gray matter, but white matter might be related, and this maturation might be related to schizophrenia onset and schizophrenia in general. However, there is a couple of different scenarios that could happen in schizophrenia, and it would be nice to have a method that tests whether it's a neurodevelopmental disorder that affects the trajectory of white matter maturation, whether something happens around the psychosis onset and then follows the same trajectory but lower, whether there's something that affects development and maturation and causes the white matter peak earlier before the healthy controls peak, or whether it's simply the accelerated aging. All these hypotheses have been, mentioned in schizophrenia literature. There was no way to test them, even with such a powerful method as diffusion imaging. So thinking or having in mind this hypothesis and having in mind the method and all the data sets that have been collected, all the small data sets that have been collected, researchers all over the world started thinking about combining the data. And there's a couple of consortia. This is as of last year or 2 years ago. There's a couple of investigators that are combining the data. This is led by Paul Thompson. This is led by people at Harvard. The Naples Consortium is led by Yale Group. One of the first consortia that combined the data and then publicized data sets is the Australian Schizophrenia Research Bank. You can still download the data, so there's a lot of data, but it is not a great resolution because it was released 10 years ago. UK Biobank is another source of acquiring right now a large amounts of data. So with this kind of big data sets, now we can attempt at least to test those hypotheses that were put forward 5 or 6 years ago. And Enigma is one of the examples of the consortium that was put together having in mind all these data sets that were collected, acquired, and stored in different places. We are part of this as well, our data. So the Enigma investigators came up with this idea that instead of sharing the data, getting together all the raw data and having all this work that needs to be done, they will ask individual sites to analyze their data and to send the results into one centralized place, and then they would just run statistical analysis. They designed the pipeline that everybody should follow, and they collected the data. They published the paper in 2018 in Molecular Psychiatry. Unfortunately, this paper, results of this paper, even though they collected almost 4,500 scans for DTI, the results were a little underwhelming. The effect sizes were on the very low end, from 0.4 all the way to 0.2, this is where we were also trying to get involved in big data analysis, and we were thinking whether it's because the data is because it comes from different populations. The clinical populations might be variable in themselves, and it might be impossible to just simply meta-analyze the data. Another possible scenario is that the data is variable because the scanners and sites are different from each other. And we started from this assumption, and we looked at some of the data that we already had. We were at that time involved in this multi-site study where we would be a imaging core, and we would be responsible for setting up the protocols on all imaging sites, so we were sure, we were trying to make sure that the protocol, the imaging protocol, is exactly identical at each site. And then we combined the data coming from matched for age, gender, and IQ healthy controls for all those sites. We analyzed, this is the whole brain or whole brain skeleton, white matter skeleton FA. And we looked at the scanners and at the different sites, and it turned out that even with the same identical sequence, very comparable subjects, you still get differences. And for GE, the differences are big. For Philips are small, but the variance is really big. So the conclusion of this experiment was that there are scanner-specific differences. Obviously, there are sequence-specific differences as well. That most of these differences cause nonlinear variation in diffusion measures. And so that gave us the idea that the harmonization of the data that is pulling together should be done as early as possible, at the as early stages of data analysis as possible. Another source of data analysis, as you can see, is just the software, different way of estimating FA, same equation, completely different results. So we've been working on this data harmonization for a while now, and we decided to use machine learning, rotation invariant spherical harmonics features to learn the differences between scanners and then apply them to then the match sort of or map the target site to the reference site and normalize or harmonize them this way. Once we were sure that this method works, and there's a couple of papers published on the performance of those harmonization methods, we've learned that we need at least 17 subjects per site to successfully harmonize the two data sets. They have to be matched, obviously, for age, gender, and IQ at least. The larger data sets, the better harmonization works, but you need at least 17. And then we combined, and obviously the ultimate goal was to try to test those longitudinal maturational hypotheses in schizophrenia. So once we were sure that our method works, we applied it to 13 independent international sites. We got the data from them, and we got the raw data, the DICOM data that we could analyze. The distribution, we're also making sure that the sites that we selected have a similar distribution of males and females, that we have all age coverage, and in both patients and schizophrenics, the age sort of distribution is the same, because we wanted to compare the trajectories, not just the means. This is in short, the pipeline that we were using. We did a lot of QCing because we wanted to make sure that we are using only very good data. Majority of those, so funny thing about diffusion is that everybody's collecting diffusion data. Not very many people know how to analyze the data, so there's a lot of people that sit on diffusion data, and it's relatively easy to get it from other people, but the quality of the data is very variable. If you don't know what to do with the data, you sometimes, or most likely, you don't also know how to collect the data, and the quality control is sometimes not there. We had to, in the process of combining and collecting this multi-site data set, we had to exclude 2 sites completely because the quality was so poor. Once the quality control was run, we ran the brain masking. We estimated the tensors using the same method, slicer. We registered everything to MNI space using ANTS algorithm, and then we used Illinois Institute of Technology Human Brain Atlas to define the regions of interest for our analysis. We did also some experiments with different atlases. There's a lot of white matter atlases. We selected this one because it seems to be the highest resolution of all the atlases available, and it worked very well for our data. And then this is what happens when you harmonize the data. So obviously this is the green is always the reference site. This is the target site that is undergoing harmonization before and after harmonization. So the goal is to bring the means close to, I mean, the signal and to minimize the differences between sites. But obviously we don't want to touch the biological variance between subjects. So we're trying not to harmonize everything. Just to remove the inter-site differences. And the green is not always the same, because the different sites have different age ranges. So out of the reference site, we were always registering or harmonizing the data. We're always selecting the healthy controls that would be matching for age, gender, and IQ, the age, gender, and IQ of healthy controls on the target site. This is also what happens when you harmonize the data and you model the data for each individual site. This is before harmonization, and this is what happens after the data is successfully harmonized. As you can see, there are some variances between subjects within the group, and those are not lost. But what we lose is this big variance right here from site to site. After data was harmonized, we calculated FA, we plotted FA against the age using quadratic regression. We tried we tried linear regression, we tried Poisson curves. The quadratic regression seemed to work the best. Then we modeled FA for each group. We looked at between group differences, and then we also calculated and compared maturational peaks for each of the groups. So again, the hypothesis that we were trying to test with this experiment were whether there's perhaps a developmental abnormality in schizophrenia that affects the entire trajectory equally, right? It starts lower, it runs lower, it peaks lower, and then it goes parallel to the healthy trajectory. Another possibility is that there is a maturation difference that would make schizophrenics peak even earlier or later than healthy controls, and that would, at different ages, create different group differences. And finally, the accelerated aging hypothesis, where nothing happens until the psychosis onset, and then the schizophrenia patients degenerate or lose myelin faster than healthy controls. And this is the actual results. These are the actual results from the data that we harmonized. For the whole brain, it looks like the differences are at every age that we are comparing those groups. Seems like the trajectory of schizophrenia patients starts lower, it peaks a little earlier, and then it deteriorates, and the differences are clear. We even estimated the percentage change at each year. It goes from 1% here, mostly because of the big variance around the ages of 15, and then it changes at the rate of 6% at the very early, very late stages of disease. And you can see that the harmonization also did miracle to the effect size. Now our effect sizes are between 0.6 and 1.6, which is way better than this Enigma results. would suggest. Interestingly, we also looked at the regional differences or regional specificity of this pathology, and we found that different tracts behave differently along the schizophrenia time course. There's a group of fiber tracts that seem to be stable, that their effect sizes of differences between patients and healthy and controls are stuck around 0.4, 0.5. There are some fibers that show deterioration at the very beginning, or show pathology at the very beginning, but then deteriorate faster. And there are those, these are the frontotemporal long association fibers. These are the limbic structures, single uncinate. And there is a third group. This is corpus callosum, forceps major and forceps minor, and they behave even, I mean, they show even bigger pathology. It starts already at the onset, at the very early ages, and deteriorates, goes with the accelerated aging hypothesis. These are the, again, results per cluster, where we show group differences at each age. And it seems that the first cluster, the limbic associated fibers, would suggest that there is a neurodevelopmental pathology in schizophrenia that does not progress. There is a group of these fibers belonging to the frontal temporal connections. They are interesting because they seem to be peaking earlier than in healthy controls. So maturation goes and then stops at some point, and then goes down again. So there's not only difference in trajectories, but there's also difference in peaks. And fractional anisotropy seems to be different at almost every, every age of the comparison, except for very early, very young ages. And corpus callosum, which seems not to be peaking at all. It seems that the maturation peak is seems to be before the age 15, and then it all shows progression, deterioration of the white matter. We also looked at peaks of maturation, and it seems that the most statistically significant peaks were showed at the superior longitudinal fasciculus, left and right, and uncinate, as well as for the whole brain, which was probably driven by those two tracts. So with this method, we can nicely now map the entire trajectory of maturational lifespan changes in FA. And what we see again is that white matter pathology is observed along the entire course of schizophrenia, that the results are consistent with abnormal early developmental lesion, but this early developmental abnormality seems to dynamically interact with maturation and aging processes, and the interaction manifests itself at different brain areas at different ages and different stages. Obviously, the ultimate goal of this experiment, this exercise, is to eventually develop some sort of normative curve where you would have, maybe it has to be based on thousands of subjects, where you would have the behavior of these biomarkers as well as maybe even other biomarkers, plasma biomarkers or other neuroimaging biomarkers, where you can say with some percent of certainty that your subject, when you scan it at the age of 2, 5, 10, 15, falls either within the normal curve, and it's safe to say that it's not at risk of developing any neurodevelopmental diseases, or it's below or above the normative curve, and somebody needs to pay attention to this subject, especially if the clinical symptoms also suggest that there might be something wrong with the patient. It would be also a tool to follow up the subjects, to monitor the efficacy of the clinical intervention or pharmaceutical intervention. So it has a lot of potential. Obviously we're not there, but we need more data, and this is the first step. Now I was telling you what to do with large data sets, but now I will also show you how one can use diffusion to learn what is underlying pathology, biological or cellular pathology, that causes FA abnormalities, that causes this pathology that we were just testing or looking at. So again, this is the same slide. It just reminds us how non-specific our FA is. We were measuring it, we were assuming and we're testing myelin hypothesis, but it's not just myelin and demyelination that causes these group differences. It could be anything other than myelination, including the expansion of extracellular space. So here, while myelin hypothesis seems to be the leading hypothesis still in the schizophrenia field, the neuroinflammation hypothesis sort of re-emerged. It was popular in 50s, but there were no tools, no in vivo tools to test it. And now, because more and more tools are available, it has been suggested that there might be neuroinflammation going on in schizophrenia and is underlying some of the accelerated aging processes, some of the processes related to the onset of psychosis. There is evidence from both cytokine increase. There is also evidence suggesting that the reduced risk of psychosis follows the treatment with anti-inflammatory drugs. And the TSPO PET ligand, which is binding to activated microglia, which is a sign of neuroinflammation, seems to be increased in schizophrenia, at least in the very early work. So there might be neuroinflammation in schizophrenia. It might be related to neurodegeneration later on. The story sort of, like the simplified version of the story goes that there's the peripheral neuroinflammation. The peripheral neuroinflammation causes the increase of blood-brain barrier permeability. The cytokines get somehow into the brain. They cause the myelin damage, or the cellular damage. The cellular damage reinforces the neuroinflammation. It's a vicious cycle, and at some point there's both central neuroinflammation and central cellular neurodegeneration. We have been working, obviously, for a long time on developing those measures that we thought might be specific to various parts of the pathology in the brain. So here, to test this hypothesis, we decided to use one of the methods that we developed in our lab. It's called free water imaging. And the simple assumption it's a one-step it's just one of the methods that obviously of the models that can be applied to diffusion imaging. It's relatively simple. It just assumes that there are, instead of one compartment of water that diffuses in each voxel, we are assuming now that there are two diffusion compartments. One is free water that is quantifying this extracellular water that might be increased when there's inflammation, edema in the tissue. And then another one, there's a tissue compartment, or there's FA corrected for free water that potentially could tell us something about tissue damage, myelin, axons, cells. There are obviously some assumptions in this model. The assumption is that diffusivity in the CSF is fixed that is a weak assumption, because if you have inflammation, you have higher temperature, and higher temperature means more diffusion or faster diffusion in the CSF. So that might be also one of the things that we're not taking into account. This method was developed not for schizophrenia and not for looking at inflammation. This method was developed to look at edema and to try to extract those fiber bundles in the area of edema surrounding the tumor. Neurosurgeons needed to know whether around the tumor the axons are damaged, and the further operation would not cause any additional functional abnormalities, or whether there are axons that are intact, they are just buried inside the edema, and they have to be very careful around the tumor. And that method seems to be working very nicely. This is before the free water was eliminated in this edema area, and this is after free water was eliminated. It was shown that there are fibers that are intact, they are just not detectable with diffusion tensor imaging. So obviously, having this method, we applied it to schizophrenia, and this is what we found. This is the population of first episode schizophrenics. These are patients that just developed schizophrenia, they're acutely hallucinating, and they've been put in the magnet and scanned with regular diffusion imaging sequence, and free water imaging model was used. And what we found was that schizophrenia patients show free water, and free water increase is in blue, and this corrected FA is in red. We found that free water is increased everywhere in the brain, in the entire white matter. This would suggest that there's something very acutely, something going on in the extracellular space that increases it, causing this free water to go up. And there is also cellular damage, but it seems to be very limited to only a few areas in the frontal lobe. The next step we did, obviously we applied it to chronic patients with schizophrenia, patients that were sick for a long time, that were stabilized, they were receiving medication for a long time. And here we still see some free water increases in the frontal lobe, but it's not anymore over the entire brain. And moreover, the tissue damage seems to be progressing from just a couple of voxels in the frontal lobe now to a little bit more than just a couple of voxels. We consequently replicated it. This is the data from Zucker Hillside Hospital in New York, where they ran clinical trials. So there were people non-medicated, acutely ill, and then we have also not imaging but clinical data on them 60 days after the treatment. And we applied free water imaging, and we saw the same thing that we saw in the first sample. We saw free water increase all over the brain, and only a couple of places where this corrected FA tissue damage occurs. And this is another not related data set. This time it comes from University of Kyushu in Japan, and it's a smaller sample, but it's a sample of chronic patients. And here we have almost no free water difference, and everything that is different between those two groups can be explained by corrected FA. So there's a lot of tissue differences. Both free water and FAT have also clinical consequences. When you look at the correlations between negative symptoms and FAT, it seems that the lower FAT, the higher clinical symptomatology. The free water is also increased in patients that hallucinate actively when compared to those that are not hallucinating. We also recently published a paper, I think it's in press, that shows correlation between free water and pro-inflammatory cytokines in plasma periphery in the Australian biobank dataset. So it seems that we have a biomarker that is finding or that is detecting consistently something acute that is going on in the brain. But is it really neuroinflammation? So obviously, free water is not the only measure of neuroinflammation. There are other measures of neuroinflammation. You can now measure cytokines in the plasma and in the CSF. These are pro-inflammatory cytokines, and they seem to be increased in schizophrenics, in patients with schizophrenia, when comparing to controls in both plasma and CSF. There's either more impressive result published in 2009 that the interleukin-1 beta is increased in CSF versus healthy controls. I've never seen this kind of results before. So it's really impressive. And then there's PET TSPO methodology that I mentioned to you before. This is the PET ligand that is supposed to go through blood-brain barrier and bind to activated microglia. Activated microglia, obviously, microglia activates when there's inflammation in the brain. And what they found was that the signal from this TSPO in the brain increases in subjects at high risk of psychosis as well as in schizophrenia patients. Unfortunately, this study was not replicated consequently by other people. There is, following this one paper published in 2016 or 15, there are 3 or 4 other papers that are showing either no changes in TSPO or decreases in TSPO signal in patients with schizophrenia compared to controls. And they are involving both chronic as well as first episode schizophrenia. This is in first episode schizophrenia study published by Jennifer Coughlin, and it shows that the healthy control, the schizophrenia signal is decreased in almost all over the brain when comparing to healthy controls. So it doesn't seem that free water is aligned with activated microglia in the brain. It doesn't look like we are talking about something very classical neuroinflammatory response. What we found looking at this Tucker Hill Hospital data set is also, I mentioned to you that we have the follow-up clinical data sets, clinical data. And what we found is that this free water imaging is correlating with successful response to medication. And it's correlating in the sense that the higher free water, the better they get after 12 weeks of treatment. So that would be contraintuitive to the inflammation hypothesis. If we're assuming that inflammation is bad to your brain and it causes the damage to the tissue, why people with higher levels of this inflammation respond better to medication. These results have also been, or have been previously reported, or this sort of results have been previously reported, and it shows that the patients, if you scan patients at the onset of psychosis and then following the medication treatment, it seems that there's a group of patients that has high levels of diffusivity in the brain. And diffusivity is sort of like it's a free water coming from the Tensor model, not from the free water model, but it's measuring more or less the same thing. If the response is strong enough, it shows that there's a group of people that have increased diffusivity at the onset that goes down following the treatment, and this is the successful treatment. And there's also a group of people that don't show free water diffusivity increase, and that doesn't change. It also doesn't make them healthier after, or less sick after medication. So it seems that maybe free water has something to do with the brain response, or mounting brain response to sorry about that. psychosis, to chemical processes that are underlying psychotic onset, and it might be a good biomarker for separating those that respond to medication versus those that might not respond to medication, or this particular type of medication. So this is basically a summary of what I just talked about. We see the extracellular water fraction that increases in first episode of schizophrenia, decreases back to normal in chronic schizophrenia. We see that all phases of schizophrenia are associated with increased interleukin levels in plasma and CSF. We see that there is a activated microglia levels may be increased in chronic schizophrenics, but definitely unchanged or even decreased in other stages of schizophrenia. The extracellular water increase might predict the outcome and the response to medication, and that the free water decrease might be also used to monitor the treatment. So that's basically it. I hope that I was able to show you how diffusion imaging can be used in both big data analysis as well as learning more about underlying biological changes. This is obviously the ongoing study or ongoing project. We started looking at PET, TSPO in psychosis, obviously without much hope, but we want to look at this data ourselves. We're also exploring other neuroinflammatory pathways that might affect extracellular matrix, but not necessarily might affect microglia. We're developing another ligand that binds to one of the elements of the neuroinflammatory pathway, MMP9, yeah, these are the grants that are funding this work, and all the people that are working on the data. Thank you.
Questions?
So I was wondering, listening to you, how many of these markers, imaging biomarkers, have been applied to other psychopathological conditions? In other words, what's the specificity of these markers?
Well, the free water, I don't think it is specific to schizophrenia. I also don't think that neuroinflammation is specific to schizophrenia. Neuroinflammation has been implicated now in depression, in mood disorders, in autism, in all sorts of, in traumatic brain injury. So it is a biomarker of something that happens in the brain. We don't even know what it is. It definitely is not specific to schizophrenia. Timing of it might be specific to schizophrenia. We've done the experiment and we published a study where we're looking at bipolar disorder versus schizophrenia, and they seemed to have an increase of free water at different stages of the disease. Bipolar disorder subjects seem to have increase of free water at chronic stages, while schizophrenia don't. So it might be something that is related to phases of the disease, to particular responses, maybe to medication, maybe to stress related to psychotic episodes, maybe to other things. Not specific.
I have a few questions. How does free water relate to diffusivity, which is also just the magnitude of diffusion?
Mm-hmm.
And then the other one's more complicated. So increased free water would imply less myelination, and yet in the mega-analysis, these trajectories were showing decreases in myelination, neurodevelopmental, aging, and so on. And I mean, you mentioned that it's still something that you're going to do in the future, look at the correlations with medication in terms of aging, but I mean, that would help to address that question.
So in my experience, first of all, the trajectories, the age-related or disease-related trajectories were on FA, and both FAT and free water would affect them. So if it's not if free water increase, it's FAT decrease. At some point they're summarizing. If they are occurring together, they would affect FA more. But even if there's no free water abnormality, the FAT is still decreased in chronic stages. So that affects this, that causes this difference in FA. What was the other part of that? Oh, diffusivity versus. So diffusivity in again looking at several different data sets, diffusivity is strongly correlated with FA. So diffusivity would follow, FA would increase, diffusivity would decrease. Free water behaves a little differently. Free water decreases at the early stages. I might have. This is on a harmonized data set. We haven't published this, but on a harmonized data set, this is sort of how free water and FAT behave. So free water would, in healthy controls, it would go down and up. In patients, it would just go up. So there's like no this developmental, maturational sort of dip, which would, I don't know what it suggests. It suggests that there's simply some pathology that happens more here than here. And FAT behaves like this. So FAT shows this progressive change. And they both together, even though free water differences are not here anymore, FAT differences are here, so that causes this FA to be at least stable, if not higher or more affected.
Oh yeah, I thought the harmonization or the normalization data you presented was really interesting. I was just curious if you see that as sort of becoming something that would be generally useful as like a pre-processing step in general, or is it more just useful in combining multi-site data?
It is definitely useful for multi-site data, but it is also useful because even if you're running multi-site study by yourself and it's not combining these data sets that were not, designed to be similar and combined together, there's still inter-site variability, and it is better to minimize it than not to do anything about it. We have one of my collaborators, Yogesh Ratty, in the lab, just got the R1 grant to do harmonization, to run harmonization on all HCP and all ABCD data. So this data will be eventually harmonized, at least with our method. Other people are working on other algorithms as well, and they will be applying them to it's not, validated or compared with other methods. So I think that there's going to be more and more of this harmonization business going on with.
On the harmonization, where is this actually acting? Is it reading in the raw data, or is it acting on the FA?
No, it's acting on the raw data, and it's reading the raw data, and it's learning the differences from the raw data and then applying it to the FA.
And have you been able to discover what those differences are, or is that sort of black box as a lot of machine learning things?
It is a little bit machine learning thing. But so your question is whether it's more related to p-values or number of directions or in field and homogeneity. I think all of this plays some role. Some of these effects are linear, some of them are non-linear, and I think it's just safer to assume that there's like a lot of noise and just remove that noise.
And your 17 subjects is when they're all just at random from the population. I guess concerns would be, what if you have a group that's kind of different ethnically or whatever, that might be at variance, and also if you were to sort of take the human phantom approach, do you think you could drop that number down to like 5 and get the same kick, or do you really need the diversity?
So one of the things that was coming from HCP data showed that the Prisma data, the data that was collected as part of the Human Connectome projects all over, are much less variable. So they are much better, have much better quality. The variance is not as big as what we've seen before. So that's a good sign. That's definitely the better data you collect, the less noise you collect, the more normalized the more similar you're. This is just one type of magnet, right? So the pulses are delivered in the same way, and the noise, if there's no external source of noise coming affecting your field homogeneity, it was supposed to be allowing you to collect similar data. NIH assumed that all the controls can be just combined together, right? So yeah.
I have another question. So how did you handle excluding data from motion in your mega analysis? This is something that we always...
What did we exclude?
No, how did you?
So we, I mean, there's a lot of computer scientists that are hacking a lot of, so, you know, we run, there's the eddy current correction, automatic eddy current correction algorithm right now, right? That like registers everything to the same space, and we looked at the outliers. Because, you know, with 1,000 of subjects, you don't necessarily want to go to each subject and slice by slice look at it. We have a software that detects the bed gradients, and we remove the bed gradients and we replace them with the neighboring gradients. And then we looked at the masks, like all the masks or every other mask that was coming out of the bed was evaluated, and one data set that we excluded was acquired with a completely different wrong diffusion scheme. So we just realized that this doesn't make sense.
So one last question. Anyone had any other questions? So while we are on the topic of harmonization, I had a question. So you said harmonization should be done at the very beginning. So maybe when the data is getting
ideally, yes.
Data. But many methods like Combat, for instance, they are doing harmonization on prospective reconstructed data. And then there are other approaches like Midas or Hydra, which are doing advanced other multivariate techniques that further account for reduced variability. So has anyone done any analysis actually comparing the results of these two approaches and seeing what?
I think Suhaila will be presenting something in Mikai, comparing Combat with our harmonization. We have a feeling that Combat, for example, over-harmonizes everything, so it just removes the biological variance.
Right.
But you know, I mean, it's a work in progress. There's a lot of people working on harmonization, so I mean, you can just criticize or show that your method is better than this other method, but can't ever win that battle.
Okay. Because when you showed the data, the scatter of the data was very compact after harmonization. So it is definitely maybe throwing off, at least in our experience, when we tried to do some data alignment, it threw off some variables out of the So let's thank Marek again for a nice